Buenos días a todos. En honor al tiempo, vamos a dar comienzo a este segundo bloque del tercer simposio de inteligencia artificial en salud. Desde el programa de inteligencia artificial en salud y el departamento de informática, estamos muy contentos de darle la bienvenida a estos bloques y en especial a nuestros oradores y keynotes internacionales que participarán en las próximas charlas. Dado que tenemos oradores internacionales, las próximas charlas se desarrollarán en inglés y recuerden que pueden hacer sus preguntas a través del chat. Okay, I will start introducing our first speakers. If you wish, you can share your screen from now. Pranav Rajpurkar is at Harvard University in the Department of Biomedical Informatics. His research approaches problems in clinical medicine with a computational lens, developing algorithms and datasets that can drive AI technologies to support medical decision-making. He co-hosts the AI Health podcast and co-edits the Dr. Penguin AI Health newsletter. He instructed the Coursera course series on AI for medicine and founded the AI for Healthcare Bootcamp program. Previously, he completed his PhD co-advised by Andrew Ng and Percy Liang at Stanford, where he also received both his bachelor's and master's degrees in computer science. Henrik Marklund is a PhD student in computer science at Stanford University and a technical mentor in the Stanford Harvard Medical AI Bootcamp. Previously, he was a research fellow at Harvard Medical School doing medical AI research. Before that, he completed his MS in computer science at Stanford and bachelor's degrees at Chalmers University of Technology, Sweden. Agustina Saenz is an associate physician at Brigham and Women's Hospital and an instructor in medicine at Harvard Medical School. She is also a clinical informatics fellow at Mass General Brigham and a Master of Bioinformatics students at Harvard Medical School. Previously, she completed her medical degree at the University of Buenos Aires, her residency in internal medicine at Einstein Medical Center, and her Master of Public Health at Harvard T.H. Chan School of Public Health. Dear speakers, thanks for being with us today, and you can start whenever you wish.
Excellent. Thank you so much for the kind introduction, and thank you for having us. I'm delighted to be sharing some of our lab research with you today, and I'm even more delighted that I'm being joined by Agustina and Henrik to be able to share this with you. So I want to start by talking a little bit about what's most exciting right now to me. So my lab aims at thinking about how we build artificial intelligence to solve complex decision-making problems in medicine. And I think of this as a goal that can be achieved by progress on three fronts. The first front is algorithm design. And here the question is how we move from these label-intensive, inexpressive algorithms that we have today to algorithms that can be label efficient and express their outputs in natural language. On the data curation front, where the state of affairs today is that we have really data that is narrow in both tasks and the diversity, and we'll talk a lot about this today, and how we get from that to data that is diverse in tasks and clinical settings. And finally, we can't discount the human piece of this, which is that currently we are really centered around evaluation of these algorithms without consideration for who is using them and how they're being used. And that's a change in the questioning that needs to happen. My group focuses in particular on self-supervised learning and multimodal learning techniques from a technical standpoint, and we focus particularly on imaging and sensors. All right, so I want to start by giving you a sense of the kinds of advancements we've seen over the past few years and where there is room for growth. So this is some early work that I was involved in, looking at chest x-rays on the left and seeing if we can detect multiple diseases with the use of deep learning. And we showed that we could perform comparably to board-certified radiologists at the detection of several diseases. And on the right, we see a single lead ECG used to monitor patients for up to 2 weeks. And using the single lead ECG readouts, can we detect abnormal heart rhythms at the level of board-certified cardiologists? And that's what we showed on the right. But both of these studies have a critical issue, which is that on an algorithmic front, they rely a lot on lots of manual labels being collected for the data. And on the validation front, they're really only evaluating on a small patient population compared to everyone we would want to apply these models on. So I want to talk about the latter piece of this and give you a sense of the kinds of things that need to be thought about when we think about data curation. It's no secret that medical data set curation is hard. It requires partnerships with hospitals. It requires IT frameworks for de-identifying the data, pulling the data, and it requires expensive manual annotation. On the front of expensive manual annotation, there have been quite a few creative approaches to solving the problem. One creative approach is when we have free text English language reports associated with the medical images that describe the images, we can mine these to be able to get labels. So for example, the unremarkable cardiomediastinal silhouette can then be cardiomediastinal silhouette can be extracted and it can be associated with the property unremarkable to get a 0 on enlarged cardiomediastinum. So we can use these labeler outputs corresponding to the images to now train a deep learning algorithm. We've used this kind of a technique to label what are some of the largest chest X-ray databases available, including a data set from Stanford Hospital called CheckSpurt and a data set from BI in Boston done in collaboration with MIT and Harvard called Mimic CXR. What we've been interested in more recently is to say, can we extract everything that is useful and important in these natural language reports? And one way we've formulated the problem is to say, can we represent text in the form of graphs? So if you see text on the left, there's some information that corresponds to just the patient information. There's some language corresponding to style, and we want to throw away all of this and retain all of the information about the content, which we do on the right. And we've applied this methodology to be able to extract these graphs which contain this content for over 200,000 reports, releasing a data set of over 6 million entities and 4 million relations. That's called RADGraph, and that's publicly available. The release of medical AI data has led to a lot of advancements, not just for chest X-rays, but also for other kinds of modalities. We've released data sets for bone X-rays, we've released data sets for knee MRIs that's allowed people to make significant advancements. I want to talk about the other piece of data curation, which is how we set up diversity in clinical settings through partnership efforts. And my first experience with this was in 2018, when we had developed these algorithms that could interpret digital chest X-rays, and we applied them to films, and particularly to photos of films. This allowed us to apply these models to the vast majority of the developing world, which doesn't use digital chest X-rays. And when we apply models to new settings, we often see there is a drop in performance. And here we saw when we applied models to these photos with chest X-rays, there was a big drop in performance. And we can tackle this. And one way we can tackle this is by introducing new data sets. So we introduced a new data set called CheckSphoto, and we collected data at scale for this data set. And then we saw that through a combination of applying new data with applying new technical methodologies, we had a big improvement in performance over the span of a year as measured on this leaderboard. So what I want to do for the rest of our time today is talk to you about an effort that I'm certainly very excited about and have been working with Henrik and Agustina on the last year, which is how do we create multi-institutional data sets for the development of robust, safe, and clinically useful medical AI models. As part of that, we want to talk to you about MED, which stands for Medical AI Data for All. So I'll stop there and hand it off to Agustina.
Thank you, Pranav. So because of how important these public data sets are in the development of AI models in medical imaging. We started a little bit over a year ago, MEDA, which is Medical AI Data for All, this CHEST consortium, where we're collaborating with institutions around the world with the goal of curating multi-institutional data sets to develop robust, safe, and clinically useful medical AI models. And to do so, we think that each of these data sets that we aim to make available to anyone to evaluate their and develop their machine learning models, that there needs to be 4 parts. One is that there needs to be a clinically useful machine learning task. There needs to be data from a diverse set of hospitals diverse in terms of demographics, equipment, so on. And then there needs to be granular, high quality annotations, and then finally fine-grained metadata. And the goal here is broadly for us to collect across a broad range of hospitals, as well as a broad range of modalities. So that includes CT, X-ray, and so on.
Agustina, you have to unmute. Thank you.
So we started with chest X-rays because chest X-ray is the most common image across the world. And the settings, we decided on 3 settings to start with. And we wanted to have a setting where we already had enough advancement in machine learning, where we knew that we could make progress, but we weren't at the point that we wanted to be in terms of be ready to for application in clinical practice. So our first setting is the intensive care unit, is the endotracheal tube placement in the unit. Our second setting is the endotracheal tube placement in the neonatal ICU, and our third setting is the evaluation for pneumonia in the emergency department. Sorry, my slides are working weird. Our first setting is endotracheal tube placement in the ICU. And why we chose this setting is, one, time matters. If we have a tube that is placed incorrectly and we don't detect it right away, a patient can have hypoxia, or we can have pneumothorax, which are very serious complications. The second reason is that radiologists are not always available. Even in places like the US, where radiologists read all the X-rays, they don't do it between 5 p.m. and 8 a.m. So there is sometimes at night, an intern may be alone, and you graduate, new residents. So we wanted to make sure that we had a support in place to help in those situations. And then there is a lack of studies with clinical relevant labels. And by this, I mean that even though there are studies where we know that we can detect an endotracheal tube, where we know if it's right or wrong, this doesn't translate into a clinical practice at the bedside, where I'm, as a clinician, I wouldn't know what to do with those type of labels. So we wanted to make sure that we were closer to clinical deployments setups. And if we look at the data sets that are collected for endotracheal tube, we'll see some things in common. So we found 5 data sets, but as you can see, 3 out of those are from the United States. Only 1 is from Spain, called PATCHEST, and 1 is from Brazil, BRAX. You can see that there are enough images. Just the amount of chest X-ray is not the issue. We have more than 200,000, for some of them more than 100,000. But as I mentioned, the labels are not where we want it to be in order to be ready for deployment in clinical settings. So for example, CLIP, which is the most similar to what we want to accomplish, are images from the NIH. And they will tell us if you have an endotracheal tube, and it will say normal, borderline, or abnormal. But if I'm a clinician and I get an abnormal label, I don't know what I do. Should I pull it? Should I advance it? The MIMIC that already Pranav mentioned a little bit, Chexpert and BRAX, have labels that tell me that there are support devices. So it means that they may be an endotracheal tube, but it just may also be a pacemaker. So I cannot do much with those labels as a clinician. And PATCHEST will tell me if there is a tube, but it doesn't say anything regarding what the placement of the tube is. And those are the barriers we wanted to overcome with this new data set. Our setting number 2 is endotracheal tube placement in the neonatal ICU. Same reason, time matters, radiology is not always available. And a difference here is that there is a lack of machine learning studies for infants. So we know that we can detect ET tubes and we can do it well for adults, but we don't know how that will translate into infants, that the x-rays are very different, especially within the first 6 months of life or when they're premature. So we wanted to see, okay, can we do this for infants? Sorry. The problem here is that there are not enough data sets. As you can see, we only found MIMIC, which has images from the neonatal ICU at Beth Israel Medical Center in Boston. And there are enough patients, but it's only one setting without the labels we need to see if we can translate this into clinical practice. The third setting is the pneumonia evaluation in the emergency department. Time matters, but for two different reasons. Not only clinically, as we know, we need to give antibiotics within the first 4 hours for better outcomes if it is indeed pneumonia. But at the same time, for workflow, a physician in the emergency room may need to see, do I need to admit this patient? Can I send this patient home? The emergency rooms are overcrowded and we need to make decisions fast. And we don't want to send the wrong patient home, neither admit the one that doesn't require it. Radiology is not always available. Many places in the world, radiologists don't read the reports, and it may be a new trainee making a mistake or new graduate and things like that. And then we have limited data sets with granular annotations. For example, most are binary. Is there pneumonia, yes or no? It doesn't tell me the extent of the pneumonia. Is it severe enough that I need to keep the patient in the hospital? Doesn't tell me the location of the pneumonia or to help me with the differential diagnosis of what type of pneumonia could it be. And we don't have laboratory values for most data sets, which, as we know, it's really important. We need to take the X-ray in context of the patient. So we need, does the patient have a fever? Does the patient have cough, shortness of breath? What are the clinical things? So those are the reasons why what we have so far is not enough to translate into practice. And if we look at the data sets, we can see that in general for these settings, we had 7 data sets, but we have similar issues. So most of them are from the US. We have the one from Spain, PAT chest, from Brazil, BRAX, and one more from Vietnam. Enough chest X-ray, again, that's not the problem, but the labels don't match amongst them. So if we wanted to do a study, we'll have to either relabel them or do something, because some of them have 14 labels, some of them have 28 labels, like VINDR, Patches has 193 labels. So it's very hard to work with that. And as we said, they're binary. And we wanted to go more and say, okay, we see a right lower lobe consolidation, or we will be able to tell the extent of how severe it is too by calculations that we will be doing. So those are the things that are not there yet and we wanted to work on.
So like Agustina said, we've been focusing on these 3 settings, and then over the last year, we have been together with our collaborators around the globe, been curating chest X-rays for these 3 settings, and we've been asking each collaborator to share between 100 and 300 X-rays. And in our experience so far, this has really made it a lot easier for more people to contribute, and a lot of people have been very excited and interested in collaborating so far. One reason it becomes easier is that, for instance, de-identification and anonymization of data is very hard to automate. So there needs to be some manual process there. And by having fewer examples rather than, in contrast to say, thousands of X-rays, that becomes a little bit more manageable. We've been doing stratified random sampling, and that just means that we ask each collaborator to select a few samples of each pathology, or both correct and incorrectly placed endotracheal tubes. And then finally, we've been collecting together with collaborators a broad set of data elements so as to enable more fine-grained evaluation. So they include demographics, equipment, metadata, and the radiology report and image. And of course, this has been there have been many challenges with coordinating many collaborators like this. And one big challenge has just been the fact that timelines for ethics review, so IRB in the case of the US, differ widely between collaborators. So it's been kind of important to just set the expectations with the collaborators from the beginning, like, hey, this will take quite a long time. And it's also been important to have like templates so everyone submits very similar IRBs. It's very hard to make updates to the collection protocol. So it's very important that one gets it quite right from the start. Finally, getting consistency in terms of collection procedure and data elements across collaborators is very challenging. And we've made like a big effort here of creating checklists and spreadsheets that help create consistency, but also recognizing that there's a limit here to how consistent people can be, and then making an effort to make sure that at least inconsistency is transparent. So we have people document as much as possible about how they collected the data, so that at least we know in what ways collaborators differ between each other.
So where are we now? We have more than 35 hospitals across Europe, South America, North America, and Asia that already joined our initiative, and they're participating, some of them in one setting, but many of them in all three settings. So I'm going to start with the first setting that we are, the one that we're working on will be closer for publication soon, is endotracheal tube placement in the ICU. So we met with a group of radiologists from across the US and other countries too, to decide what were the landmarks we needed to have in order to be this as objective as possible and decrease the amount of inter-rater disagreement. And everyone was in agreement that we needed to mark the two medial end of the clavicle and the carina, and obviously the tip of the tube. But the carina alone wasn't enough. And we'll have 10,000 images from those public data sets that I mentioned priorly from the different countries. And then we have already more than 2,000 images from our partners that we will relabel all of them by board certified radiologists. So our model has two different types of outputs. One is for the radiologist. So we're gonna help with radiology report filling. So you can see in the left, this is how an report in the US look like. And what it's in bold is what the model will generate. So it will tell you if it's correct and where it is. And then possible outputs you can see on the right. Let's say the tube is in the right bronchi, it will tell you pull the tube. So this will overcome the challenges that we said before. It's like abnormal. Here we'll know exactly where it is located and what we should do with that output. So we'll help the clinician directly at the bedside and the radiologist with the report generation. And again, this is for the clinician. We can see in the image, the tube is on the right main bronchi. The radiologist may not be in-house, or even if it is in-house, they won't read the report within 10 seconds. So as soon as the image is taken, there is a message that go right to the bedside. So the clinician, even though may know how to read the report, may not read the x-ray as soon as it's taken, maybe doing something else with a different patient, waiting for the radiology to be radiograph to be ready. So this will help decrease the amount of time until we recognize that the tube is misplaced.
And the way we've set up the machine learning task is that the machine learning model is trained to detect the location of the carina, the ET tube tip, and the clavicles. And then we can use those locations to compute the distance between these landmarks, because we have estimates of how many pixels are between the landmarks, and we know roughly how big a pixel is in terms of millimeters. And there are two additional benefits of doing landmark detection here compared to classification, apart from being able to give a more fine-grained output. It's that when you have this more fine-grained output, you can do more fine-grained evaluation. Rather than saying, was it correct or not, you can say, how correct was it, or how bad was the prediction? And this is very important, because oftentimes what matters is just how wrong you were. And if you're a lot of wrong, that's a big problem. But if you're a little bit off, just a few millimeters off, that may not be an issue. Further, there's reason to suspect here that you will have a lot less spurious correlations, a lot less sort of distracting features when you're doing landmark detection compared to classification. And therefore, we expect much better generalization by setting up the task this way. And then finally, our overarching research question here will be to study how well do these machine learning models generalize to new hospitals, regions, and equipment.
Oh. So if you want to join our initiative, there are many ways you can do so. You can collaborate in one of our 3 settings. We're still recruiting hospitals to participate, either ICU, emergency room on the neonatal ICU, be part of one of our prospective trials. We're going to start prospective trials very shortly with partners in the US and outside of the US. And as I mentioned, in terms of like the radiology report generation and the workflow to the clinician. Or you can share new ideas for our next project. As Henrik mentioned at the beginning, we want to expand to other sorts of images. And because we already have this consortium and this more than 35 partners, it will be easier to reach out and get more images. So it's something that we can work and escalate faster because we already have all those connections. So you can email us to chessconsortium.listserv.med.harvard.edu, or you can also check out our website and sign up right on the website, and we'll get back to you also shortly after that.